All right, I just wanted to go through the answers to Assignment 6b to make sure that you were clear on all of the details of each answer. So again, some of these are pretty straightforward. So like number one, what does SPAC stand for? That's a soil plant atmosphere continuum. And that's important just to remember that the water column, we think of it as a column going from the soil up through the leaf, is a continuous column, and then that's also being pulled up through the plant based on the relatively dry air of the atmosphere. So that's just all one continuous movement of water molecules through the plant. What starts that process, and that's again the relatively dry air in the atmosphere. And because we're describing the water status in a system by the water potential. The water potential in the atmosphere then is really low because we know water is moving from places of high water potential to low water potential. So the atmosphere is really low water potential because there's very little water there relative to what's in the plant. And so that dry atmosphere causes evaporation from the leaves inside the leaf. And you can almost think about it like dominoes in reverse, right? You lose a water molecule in the leaf, and so the water column has to move up a little bit to fill the void where that water molecule left. Next water molecule gets evaporated, water column moves up again. But of course that's happening to millions of water molecules all the time. Okay, we discussed two things that will lower water potential in a plant. So we're thinking, specifically in a plant now. And those two things are tension on a water column. So as the atmosphere is pulling those water molecules out of the leaf, that's kind of causing that water column to be under tension. If the soil dries out at the same time, then you've got the atmosphere pulling on the water column at the leaf end, and then you've got the soil water kind of like pulling on the other end. And so as those two things become really dry, the soil water potential and the atmospheric water potential, that causes a lot of tension to be on the water column and reduces the potential energy in that water and makes it more negative. And then if you add solutes, that's going to cause those molecules to stop moving so much, and that reduces the potential energy of water again, and would make it water potential more negative. Then water potential values in plants that we work with are always negative because pure water with no solutes and no tension on it is the reference point. That's zero. And so anything that reduces the potential energy from there is going to make it be negative because it's going to be less potential energy than that pure water that's not under tension. So that's why we're always dealing with negative values. The figure below shows the relationship between water potential of branches of a tree and the conductance of water of those branches. So the question is, what's happening inside the branch that's causing it to decline as water potentials get more negative? Or remember, as the water potentials get more negative, the tension on that water column is greater. And so what's happening here, of course, is that there are embolisms forming inside the branch. So that's when water gets pulled into a vessel element or a tracheid. That water bubble, air gets pulled in, and that air bubble expands as soon as it gets in that high tension of that water column. And so then it fills with air, and then that cell is no longer able to conduct water. And so if this happens to a couple of cells, which happens every day, there's embolisms every day in a plant, but if it only happens to a few cells, it doesn't really affect the conductance of water through that branch or through that leaf. But if you start getting lots of these embolisms show up, then it starts really blocking the movement of water and can cause a reduction of water movement to the leaves. Okay, so why would really low water potentials be bad for a plant? So low water potentials can result in cells losing their turgor. So if the turgor is lost, then that cell basically collapses on itself because it relies on there being enough water in the cell to maintain its structure. So when we lose turgor, that's when we also see wilting of the leaves. But also when that turgor is lost, a lot of the functioning of the cell just shuts down. So there's lots of stuff going on in the membranes of those cells, and when they collapse, the membranes just aren't able to maintain that same function that it had before, and other things kind of cascade from there. So losing turgor is one thing, and then those embolisms that we just saw in that last graph. And that's important because no longer is water going to be able to be conducted through that vessel or that trachea. Okay, so the two pictures below represent two plants that they experienced a freezing event. And when freezing events occur, remember, there are always air bubbles that form inside the cell, but they don't necessarily embolize the whole cell as long as that air bubble can diffuse back into water as that water thaws out. So the question is, which condition is more likely to lead to an embolism once the water inside the xylem thaws? And the correct answer is plant B, because if you see the water potential of the soil, there's more tension on that water column. And the more tension there is on the water column, the harder it is for that air bubble to diffuse back into water, or dissolve back into water, I should say. So if there's tension on the water column, it's more likely that bubble is going to expand when the water thaws and fill that vessel with air and preventing it from conducting water. All right, so I ask you to calculate the conductance of two different plants. So then the question is, which one invests more carbon in their xylem, and how did you come to this conclusion? So let's just look at these numbers, and the actual calculations aren't quite as important, but it's helpful to see those final numbers. So plant A is going to have a much lower conductance than plant B. So you can see there's a whole order of magnitude difference in conductance. This one's 10 to the minus 14, this one's 10 to the minus 15, if you did all the same unit conversions I did. But you should see an order of magnitude difference regardless. But look at plant B. It reached this conductance by producing 20 more vessels than plant A. So the vessels are actually a significant cost of carbon, because the lignin of the vessels is a material or a molecule that's really hard for the plants to produce and requires a lot of carbon. So if a plant's going to produce, it's going to make 30 vessels to, oh sorry, I got backwards here. Oh no, I'm fine. Okay, I forgot what the question was. Which one do you think would invest more carbon? So the one with 30 is probably going to invest more carbon, even though, I guess here's what I should say, even though they're smaller. So here's the radius of the two vessels. So this one's half the radius, but it's produced a lot more. And if we did the calculation on total volume of cell wall area, it's going to be a lot more in this one that produced 30 vessels. So just the number of vessels is really kind of what dictates how much carbon a plant invests in the xylem. And then the second part to that is, how did you come to that conclusion? And so what you would really be doing is looking at the number of vessels. So that's captured in the answers here. I should just read my answer because it said it more clearly than I just did. Okay. Question 9 asks, which of the plants for number 8 would you expect to find in northern Canada, and why? So we look at the structure of these vessels, or the xylem. This plant B has much narrower, half as wide of vessels as plant A. And if you remember, the narrower the vessel, the smaller the air bubble that is formed when it freezes. And the smaller the bubble, the easier it is for that bubble to diffuse back into water when it thaws. So we would expect plant B to be found at more northern latitudes because it can handle freezing events better than plant A. Okay, so number 10, based on the data presented below, calculate specific root length of each plant, and then tell me which plant you would expect to take up more nutrients more quickly. So I gave you the mass and the specific root length. So this is specific root length, so it's length of the root system divided by dry biomass of the root system. And so if you do length over mass, you'll get 61.5 for plant A and 43.2 for plant B. So plant A has a higher specific root length, so we would expect that one to have per unit biomass invested, it's going to have more root length in the soil, so we would expect it to uptake nutrients faster. Now notice that plant B has more root length than plant A, but it had to invest a lot more carbon. So we would expect, even though it might have more root length in the soil, we would expect this one to be doing kind of its physiology, and uptake tends to be a bit slower, and it's not as thoroughly exploring the soil as plant A. So we'd expect plant A to uptake nutrients more slowly. We could be wrong based on specific root length, right? This is a metric that generally captures this pattern, but it's not always correct. So plant B could take them up more quickly, but we would expect plant A to. All right, based on the data below, which of the two plants do you think occupies sites with high soil fertility and why? You probably know that nitrogen deposition is occurring throughout the world, or at least especially near urban areas, and so how would you expect the relative abundance of these two species to shift in the future? And I probably should have said, you know, a good way to think about this is, let's say right now these species are about 50-50 in a community. So they each make up about half the plant community. So then the question is, down the road, how would that change? So plant B clearly has a more conservative nutrient use strategy, and we can tell that by both how long it holds on to its leaves and how efficiently it resorbs nutrients from its leaves before they're senesced. So it's able to extract a lot more of the nitrogen before it drops its leaves, plus it keeps its leaves for 2 extra years. Now we would also know that this plant, or we would expect this plant to have higher LMA values so that those leaves could last for a full 5 years versus 3 years. So a plant has to invest a lot of carbon in the leaf structure, but then if it lasts 5 years, it can reuse that leaf over and over. So it's going to be more conservative with nitrogen use. But if nitrogen is being added to the system and becoming less limited, then we would actually expect plants that have a more liberal use of nitrogens, or less conservative use of nitrogen or nutrients, to be more successful in that system. So over time, as this system had nitrogen added to it, we'd actually expect plant A to do better in that particular ecosystem. And so that would change, you know, the function and the structure of an ecosystem because of that nitrogen deposition. And indeed, we're seeing this happen in some places where as nitrogen deposition becomes a major contributor to nitrogen in the soil and making nutrients sometimes non-limiting, we're seeing plant species shift due to how they utilize nitrogen. Okay, that's it for assignment 6b. So again, corrections to these are due by the end of the week. And hopefully just doing these corrections and thinking through them are going to help you do better and quickly find answers on your exam.